Hi, so today I'm going to be starting the first video of my new biology series. This is essentially a continuation of the old biology series, but I'm going to now be discussing not only content from the grade 12 curriculum and the IB curriculum, but also a lot of content for the International Biology Olympiad. Now, this is part because I want to prepare for the Biology Olympiad myself, and also because I feel like the content behind the Biology Olympiad, there's not enough videos out there that cover this content. So I was thinking that it's probably a good idea for me to start. So the textbook we're going to be using is Molecular Biology of the Cell by Alberts. So right here. You can find that online just by doing a quick Google search. I'm not going to put the link due to copyright reasons, but you can probably find it yourself. Right, so we're going to be starting with the second chapter. So we skipped the first chapter because a lot of that is just an overview of what's actually in the entire subject of biology. This particular series is probably going to focus more around biochemistry, but this first video, there's a lot of overlap between the grade 12 curriculum and the biology Olympiad curriculum, so this is going to be a combined video. Right, so let's start with, I'm just going to turn this in read mode. Oh, whoops. Hold on. Give me a sec. Right. And then read mode. Right. So the first part is just an introduction. So it says that three big unifying themes of biochemistry is first that it relies heavily on carbon compounds, that it always occurs in an aqueous solution, and third, that it is incredibly complex. And this will become clearer later. In biochemistry, the vast majority of molecules are made up of C, H, N, and O, so carbon, hydrogen, nitrogen, and oxygen. Phosphorus and sulfur, which make up the entire Shinop series, are the molecules that are the atoms that are the elements that are quite important in the grand scope of things when you consider proteins. Down here, we can see the main elements in cells uh, made in a graphically appealing manner. So, uh, anything colored in red is extremely major. It's almost ubiquitous. The ones highlighted in blue are kind of uh, also kind of ubiquitous, but they're not as ubiquitous as the four red ones. Um, the yellow ones are kind of iffy. We don't really know if they're actually essential for humans. Um, and the green ones are just the general elements that are required in trace amounts. So stuff like iron, nickel, manganese, molybdenum, zinc, selenium, those kinds of things that you do need, but not as much as even the blue chemicals. Right. So it covers covalent bond. The textbook covers covalent bonds. Now, covalent bonds are essentially, if you guys haven't gone to a basic chemistry course, covalent bonds are the type of bonds that link atoms together to form molecules. So these bonds are stronger than the. These bonds are generally stronger than no, these are definitely stronger than non-covalent bonds, which are the intermolecular forces such as dipole-dipole forces, and so kind of like two magnets being pulled together. Let's say you had a positively charged hydrogen on a water molecule and a negatively charged oxygen on another water molecule, then the positive will attract towards the negative. It's kind of like that sort of attraction. Um, Now, covalent bonds can sometimes be weaker than ionic bonds, but just generally speaking, covalent bonds are the stronger bonds of the two. Now, as I was talking about hydrogen bonds are really important because remember the second point I talked about in the beginning of the video, that everything always occurs in an aqueous environment. So, life on Earth begins in the ocean, and the reason that it does begin in the ocean is because, well, no, it's because it begins in the ocean that all life on Earth is adapted for an aqueous environment. And so, therefore, cells really require the chemical properties of water. In each water molecule, the two hydrogen atoms are linked to the oxygen atom by covalent bonds. These two bonds are superpolar because the oxygen is strongly attractive for electrons. This is known as electronegativity, whereas the H is only weakly attractive. If you want more information on this, check out our chemistry videos on Info Index. Anyway, there's an unequal distribution of electrons within this water molecule, and uh, as a result, there is a net positive charge on the hydrogen atoms and a net negative charge on the oxygen atoms. When the positively charged region meets a negatively charged region, you get an electrical attraction that is known as a hydrogen bond. These bonds are a lot weaker than covalent bonds. I have discussed this before, and are easily broken down by the random thermal motions, so kinetic energy, because heat is just shaking molecules, the reflected heat energy of the molecules. Uh, Thus, these hydrogen bonds only last for a short amount of time, but if you have a lot of them, they can ultimately form a really strong network. For example, water molecules can form hydrogen bonds between each other to create a network where these bonds are continually being broken and formed. So due to these hydrogen bonds, water is a liquid at room temperature compared to a lot of covalent molecules, which are gases because they don't have these same intermolecular forces. Now, stuff like alcohols. So anything with an oxygen and an OH, a hydroxide group, and hydroxide group, hold on. Hydroxide group basically is just this thing, can interact with water. So anything in general, anything that is with that is polar. So something similar here where there's an unequal distribution of electrons can interact with water. Now, water is special because hydrogen bonds have to occur between like oxygen or nitrogen and hydrogen, but anything that is negative in general can kind of do dipole-dipole forces. But let's focus on hydrogen bonds. Alcohols can do this because they have a very electronegative oxygen attached to a hydrogen. And so the mechanisms that we just explained back here are active. So many of the so these things like alcohols are very common in the environment of the cell. So sugars, DNA, RNA, and most proteins are able to hydrogen bond with water and form those big networks. Hydrophobic molecules, like fats, are not charged, and so they can't form hydrogen bonds and do not dissolve in water. Hydrocarbons, such as fatty acids, are an important example because they are linked to carbon atoms by a largely nonpolar bond. Remember, carbon and hydrogen have a very similar electronegativity. Thus, cannot form effective hydrogen bonds to other molecules. This makes hydrocarbons as a whole hydrophobic, a property that is exploited in cells. And we'll see this with cell membranes. More on that later. So there are four major types of non-covalent attractions, including hydrogen bonds, that help bring molecules together in cells. So these four are the electrostatic attractions, also known as ionic bonds, hydrogen bonds, van der Waals attraction, and hydrophobic force. Now, hydrophobic force isn't really a force. Hydrophobic force is just when you have a bunch of uncharged molecules within a charged solution, the uncharged molecules want to aggregate with each other because all the charged molecules are binding with one another and not binding with the polar one. So it gets isolated and it's just more energetically favorable for the isolation to be together. Imagine like a bunch of loners in a group of like social people, and then the loners stick together, right? Because they don't fit in anywhere. So, that's what hydrophobic force is. Van der Waals attractions are just generic London dispersion forces, dipole-dipole forces, those kinds of things that are what you usually think of when you think of uh, intermolecular forces. Now, individual covalent attraction is uh, a l- now the properties of four non-covalent attractions are presented in the panel 2-3. So panel 2-3 is just somewhere around. Wait, where is it? Okay, apparently. Okay, hold on. Oh, I can read. Hold on. This what? What? Or whatever. Apparently, panel 2-3 doesn't exist. But figure 2-3 doesn't exist apparently. But that's weird. But the table down here really compares the non-covalent forces and their overall length in the bond. So ionic bonds are not as strong, so they're gonna be longer. Hydrogen bonds are even weaker than ionic bonds, but they are an intermolecular force, and as a result, that is quite impressive for an intermolecular force. Van der Waals attractions are basically everything else, and they're not as strong. Sorry, give me a second. Sorry, I just had to do something. All right. Back to intermolecular forces. So, hydrogen bonds are the strongest of the intermolecular forces, other than ionic and ionic bonds. In non-covalent forces, they're the second strongest. So, um, a typical hydrogen bond looks something like this. So, you have a highly electronegative atom attached to a hydrogen, and a highly electronegative other atom that is negatively charged. So hydrogen is less electronegative than nitrogen. You have to have hydrogen for a hydrogen bond. If it's not hydrogen attached to a highly electronegative atom, it's just dipole-dipole. So anyway, so hydrogen is attached to a highly electronegative atom, and the positive end moves towards the negative end to form a hydrogen bond. So that's what a hydrogen bond is, and yeah. Uh, the rest of this blurb just talks about how all the interactions work. Again, hydrogen bonds are super important. They need to be in a straight line for maximum effect, like shown in this diagram. And this just talks about how hydrophobic force really isn't a thing. It's just loners sticking together. Now, polar molecules form acids and bases in water. So, um, what this is talking about is basically um, what happens um, uh, when a when a highly polar covalent bond is between hydrogen and another atom is thrown in water, and hydrogen is just thrown around in the solution. So it says here that the hydrogen bond, the hydrogen atom in such a molecule gives up its electron almost entirely to the companion atom, and so it's basically just this hydrogen nucleus, in other words, a proton, um, that is just floating there. So when this proton is just floating there, like kind of, if it's gone, like no one really cares. So right here, this is an acid-base reaction. The hydrogen is floating here, no one really cares about him. And so water is like, hey, I'll take care of him for you. So that's really an acid-base reaction. There's a hydrogen atom attached to a very electronegative other atom, and when it interacts with water, water just takes the hydrogen. That's when water is a base. When water bumps into another atom that is another atom that is a stronger base, or in this case, another water molecule, and it decides, hey, I don't really want this hydrogen anymore, then it just bounces it right off. Now, the reason that well, you you will never see just a lone hydroxyl just floating here. It's usually going to be from a part of a bigger molecule. But basically, what's going on here is that the hydrogen is taken from this molecule, and he becomes negatively charged because he no longer has one of the protons that was present in the overall molecule. You're not going to go any lower than this. the max you can go down is like one hydrogen for water. Some other things known as polyprotic acids or bases uh, can donate more than one hydrogen or take more than one hydrogen, and this is just because of the inherent qualities of the molecule. So anyway, this just explains how the acid-base reaction happens. When the polar molecule is surrounded by water molecules, the proton is attracted to the partial negative charge on the oxygen atom of the adjacent water molecule, which forces the hydrogen atom to leave and stick to the other other water molecule. And sometimes it just bounces back and forth because, well, if you can take a hydrogen atom from me, then I can take hydrogen atom from you. So it just floats around in solution. The substances that release protons are termed acids, while the ones that accept ions are known as bases. So the higher the concentration of H3O plus, so this is what happens when hydrogen is donated to another person, the more acidic the solution. So H3O plus is present in pure water at a concentration of 10 to the power of minus 7 molarity. And this is known as the Ka, the the Ka of it's not the K, sorry, the uh, the Kw of water, which is just the general concentration of water in the solution. Um, so by convention, the H3O plus concentration is known as the hydrogen H plus concentration, and is expressed using the pH scale. So the pH scale is a logarithmic scale that it's the negative logarithm of the concentration of H3O plus ions. So let's say you had 10 to the power minus 7. So what's the log base 10 of 10 to the power minus 7? That's just negative 7. And then if you were to take the negative of that, that would just be 7. So that's a pH of 7, and pH of 7 is just pure water, as you can see here. And it's the basic pH of completely neutral water, and if you throw an acid or a base in there, that's going to change. So acids are either strong or weak. Strong acids donate their protons super quickly and never take them back. So you know how like water is kind of iffy here? It's like it takes the hydrogen ions and it's like, I kind of want it back. So then the reverse reaction happens. That's why the equilibrium arrows go back and forth. Well, hydrochloric acid doesn't do that. When hydrochloric acid donates the hydrogen ion, it's just like bye. Okay. Um, Weak acids are like the water over here, just kind of clingy, it just takes it back when it feels like it, right? Um, the again, opposite of an acid is the base. Sodium hydroxide is basic because it dissociates readily in aqueous solution to form sodium plus and hydroxide ions. And so, because of this property, sodium hydroxide is a strong base. So, again, strong bases and strong acids are sort of um, uh, used in a how would I say this? Used in a lab chemistry setting more than a biology setting. In a biology setting, you usually use weak bases and weak acids. These are important because these guys essentially regulate the pH of the water by either taking or releasing hydrogen ions whenever they're present. So uh, there's always a balance between them, and so they work as something known as a buffer, where they basically keep the pH at a certain level. So it talks about buffers here. Um, so starting from the very beginning of the video, we talked about how carbon compounds are very important. So there are a few macromolecules. There are a few macromolecules, but these macromolecules themselves have a lot of things known as functional groups that are important components of the molecule. Right. So um, this blurb just talks about how carbon is really the sort of backbone of all the organic molecules. So right here, while everything else is said to be inorganic. So, as I was saying about functional groups, stuff like CH3, hydroxyl, carboxyl, carbonyl, phosphate. That's not phosphate. That's phosphate. I don't. This should be changed. Should be PO4. Um, the sulfhydryl group and the amino groups, these all sort of occur repeatedly. I think the reason why this is drawn this way is because the oxygen it's bound to, the other oxygen, is part of the overall molecule. Anyway, phosphate, everyone knows that phosphate is PO4 2 minus, but it's often drawn as PO3 2 minus for whatever reason. So anyway, these groups are known as functional groups. They basically do all the hard work in the molecule and basically mediate the interactions between the molecules. Pretty straightforward. So cells contain the four major families of small organic molecules. Again, I've sort of hinted at this, but these guys are known as macromolecules. Four macromolecules are proteins, nucleic acids, large polypeptides, and large polysaccharides, and um, lipids. Sorry, it's just left me. So these four are the big ones that you should know. Uh, these small molecules often have more than one role in the cell. They sometimes act as a potential subunit, or they also act as energy sources. So small organic molecules are a lot less abundant than the organic macromolecules. Um, so when it was talking about small organic molecules, it was just basically saying the parts of the macromolecules. You can think of them as basically under the same subcategory, except macromolecules are the polymers, while the small organic molecules are the monomers, the ones that make up the big cells, like the big molecules like DNA being made up of nucleotides or something like that, right? So it says right here, the four main families are amino acids, nucleotides, fatty acids, and sugars. You can also just memorize the macromolecules and just think of the monomers under that category. So these four families of small organic molecules account for a huge amount of the cell mass. Amino acids and proteins are especially amazing, and I'll cover that in a next video. So macromolecules are probably the most abundant carbon-containing molecules in a living cell. So there are the building blocks that make up the cell and are also the components that confer the most distinctive properties of living things. Macromolecules in cells are polymers that are constructed by linking the small organic molecules into a bigger thing. So if you stick a bunch of amino acids together, you get a protein. If you stick a bunch of sugars together, you get a polysaccharide or complex sugar. If you stick a bunch of fatty acids together with a glycerol, you get just the fat, a generic fat. If you stick a bunch of nucleotides together, you get DNA or RNA. This is RNA, not DNA. Since you could look at the carbon here, it has the oxygen, whereas in deoxyribonucleic acid or DNA, you don't have this oxygen. So this is another example of emergent properties. These super long polymers are made up of these tiny guys that you wouldn't otherwise be able to guess their function. Um, Now, again, proteins are really, really cool because they can fold and they can basically shape themselves to whatever purpose they're supposed to do. Many of them serve as enzymes, and enzymes basically catalyze all the reactions that are happen in the cell, whether that be extract energy from food molecules or fix carbon dioxide into a sugar, or even to build components of the cell, or even make other proteins. Well, kind of. I mean, technically, ribosomes are ribozymes, make it made up of RNA, whatever. Uh, Enzymes are really cool, and they're the result of proteins. So, the chemical reactions for sticking them together uh, ultimately have a lot of important features in common. They occur through a condensation reaction, where basically you have a hydroxide, you have a hydrogen, or another hydroxide. Ooh, I can make water because you have enough atoms, and then the water is created, and the two molecules are stuck together. So you create a water, you lose a water, and then these two guys are stuck together, and then you keep sticking those guys together until you get a longer component. Uh, s- Apart from some of the polysaccharides, most of these macromolecules are just made up of a bunch of limited monomers. Uh, 20 different amino acids assemble proteins, which is just an example of that. So it's important, it's super important that the chain is assembled in a very precise sequence. Uh, I'll get into this in how this happens in a later video. So the non-covalent bonds are super important in deciding the precise shape of the macromolecule. This is especially true for RNA and for proteins because their conformation really, really matters. Like with sugars, like you're just the energy form, so I don't really care that much how you're shaped. I mean, sure, cellulose is a straight chain, and that's why it's so strong, but all the other sugars, like I don't really care. But with proteins, so polypeptides, and with RNA, these guys have to have really good non-covalent bonds to decide how they're going to look, right? Think of DNA, think of the double helix. This is because of non-covalent bonds. There are hydrogen bonds going between the components of the DNA molecule that give it that double helix shape and make sure that it doesn't just break apart and turn into one stranded, uh, a single strand. So these, this just talks about how non-covalent bonds can be so strong. If you get enough of them, then they can cause the chain to rotate and become a certain shape. Uh, yeah, so um, this type of binding basically underlies all biological catalysis. Uh, Biological catalysis is basically just talking about what the enzymes do. These non-covalent bonds basically facilitate everything. They make sure that the shape of the protein is correct. They make sure that the everything fits together, that the stuff that's supposed to go with another guy is going together. So I don't know how much you guys know about enzymes. Basically, enzymes have an active site, which is sort of like an area where it's sort of like a little pocket where whatever the enzyme is trying to catalyze, that's where the pocket thing goes in. And non-covalent bonds ensure that the enzyme just doesn't catalyze any random thing, it catalyzes a very specific thing. Um, Binding of this type, so they also allow them to become the building blocks for the formation of larger structures, such as multiple things sticking together. So, see this strand, right? Let's say this is a polypeptide, this is a polypeptide, this is a polypeptide. It's sort of disorganized, but once you throw non-covalent interactions together, like dipole-dipole forces, hydrophobic interactions, electrostatic attraction, and hydrogen bonding, um, you start to get sort of a shape whenever they start these disorganized polypeptides start working together and binding to each other non-covalently. So that's it for this video. The summary here just says that living organisms are autonomous, self-propagating chemical systems that form a restrictive set of small carbon-based molecules that are essentially the same for every living species. Now, each of these small molecules is composed of a small set of atoms linked to each other in a precise configuration through covalent bonds. The main categories are sugars, fatty acids, amino acids, and nucleotides. Sugars are a primary source of chemical energy for cells and can be incorporated into polysaccharides for energy storage. Uh, so basically, it's just saying like sugars are the small organic molecules that make up polysaccharides, which polysaccharides is just a bigger molecule, the polymer. Fatty acids are important for energy storage, as well as the formation, and most importantly for the formation of cell membranes. You guys should probably already know this. This is the result of they're talking about phospholipids. Long chains of amino acids form the versatile macromolecules known as proteins. Partially true. Proteins can be made up of multiple polypeptides. Amino acids are just the small organic molecules of the macromolecule polypeptides. Uh, Nucleotides are important in energy transfer, while also serving as the subunits for informational macromolecules RNA and DNA. So just remember in this picture, to summarize it pretty well, um, these are the subunits, and the bigger units are, when they stick together, form an emergent property and have their own properties independent of the original subunit, and that cells take advantage of that, basically, uh, uh, in the cellular environment. Most of the dry mass is just macromolecules that I've just been discussing this entire video. Uh, Most of the protein molecules, many of the RNA, is folded to a unique conformation that is determined by their sequence of subunits. So this is just talking about the non-covalent interactions of the protein molecules that, or not even protein molecules, but the polypeptide molecules, and how a bunch of these non-covalent interactions can force a conformation. So something like this. This folding process creates new surfaces, again, emergent properties, and it depends on a large set of weak interactions that are produced by non-covalent forces between atoms. So that's it for this video. Thanks for watching, and I'll see you next time.